Hello students, in today's class we will be studying about sterilization or disinfection. We will be studying about the terms that is used in this process. So to begin with, basically we know that microorganisms, what is microbiology? Study of microorganisms, and they are very small. It has to be magnified and it is studied, it is learned with the help of a microscope. What is germs here? Germs, basically we can say germs are the ones which are pathogenic, or germs are the ones which are harmful to mankind. Now, is it necessary to control these germs, or is it necessary to control the harmful microorganisms? Now, why are they harmful? Why is it important to control the microorganisms? There are various reasons. Now, let us see these reasons why we need to control the growth of the microorganisms, or why do we need to kill these organisms? To prevent the spread of diseases, to prevent the transmission of diseases, probably from plants, diseases. You know that microorganisms can cause diseases in plants, in animals, in man. So these microorganisms are the ones who are responsible for these diseases. So to control them, to control the spread, we need to control the growth of the microorganisms. To control the growth of unwanted microbes in industrial production. Suppose I am working in an industry and I am producing a particular component there. If there is unwanted organisms growing, it may interfere in the product. So definitely to prevent the growth of unwanted microorganisms in the industrial production, the microbes can spoil the food as well. So to prevent the spoilage of food, I need to control their growth. In order to avoid contamination in pure cultures, I need to preserve my cultures in the pure form so that I can study it later on. I can use it for various purposes. So in order to prevent these from getting contaminated, I want the organisms in pure form. So unwanted microorganisms needs to be controlled, and growth of unwanted microorganisms needs to be controlled. Also, in my research work, suppose I am using a particular organism and I want to know what is the result of that. So if there is contamination, if the organisms are not in pure form, definitely the results also is going to be hampered there. So in order to prevent contamination and in order to prevent getting a wrong result, I need to control the growth of microorganisms. So these are the various reasons why I have to control these germs or these microorganisms or these harmful organisms. Next, how will I control them? There are various methods that I can control them. I can either kill the organisms or I can inhibit the growth. Now, what is the meaning of inhibition? Inhibition meaning I am not killing the organisms here, but nor am I removing them, but the organisms are there, but the growth is becoming stagnated. Next, I am eliminating them. I can eliminate them, meaning I will remove the organisms from that particular area. I can reduce the growth. There, meaning to say, probably the load of the microorganisms is huge. So what do I do is, I apply a chemical or I use a particular technique so that the load becomes less. Because the load becomes less, it is not in the danger level anymore. So the load is, I reduce the load. Either I will remove them or I will kill them. I will reduce the load. Okay. So these are the various methods. I can either kill, inhibit, eliminate or reduce the load. Now what do I do to do this? I will use something called as antimicrobial agents, or I will use certain physical methods for controlling the growth of microorganisms by various techniques there. Now what kind of agents do I use? It depends on the how much time do I have to get rid of these microorganisms, what kind of microorganisms do I need to get rid of, what stage they are in their growth, and also where they are present. So all these things will, based on all these criteria, I will decide which method I will follow. Next, we need to study about some terms. As I had told you, the first term that we will be studying is asepsis. Now, what do you mean by asepsis? Asepsis, it is the opposite of sepsis. Now, what is sepsis? Sepsis is basically growth of these harmful microorganisms, okay, on living tissues, and these organisms on the living tissues will result in something called as putrefaction or rotting of these living tissues. Now, that is called as sepsis. If these microorganisms, harmful organisms, grow on living tissues, they will result in something called as putrefaction or rotting. Now, that is called as sepsis. Now, avoiding them from sepsis, or eliminating or just preventing these organisms from causing sepsis, is called as asepsis. That is removing, just not allowing them to enter into this particular condition. So, that is called as asepsis. Moving on to the next term, that is antiseptic. Now, what is antiseptic? It's basically an agent that I will use. It could be a chemical, okay, and this chemical is used on animate objects, okay, animate objects. I will use this either to kill, inhibit or reduce the growth of the microorganisms. These antiseptics, they will kill the vegetative cells, but they will not destroy the spore formers. Antiseptics, when I use, it is as I told, it is used on animate objects. It is not toxic to the animate objects on the living tissues. At the same time, it will cause minimal damage to the living tissues, and also it can kill the microorganisms or get rid of the microorganisms. This is antiseptic. Moving on to the next term, that is sterilization. Sterilization means killing or removing all the microorganisms. It is zero organisms is called as sterilization. No organisms in a process. I get zero organism in that particular area or in that particular place. Now, that is called as sterilization. Now, I can get this by killing the organisms or by removing the organisms. Now, this can be attained by a chemical, generally by physical methods. I will obtain this. Okay. Moving on to the next term, that is disinfection. Now, here disinfection meaning I will use this term disinfection by using an agent that is a disinfectant. What is a disinfectant? Disinfectant is an agent which is used to kill the microorganisms, but definitely not all. They are used to kill the vegetative forms and the non-resistant forms. The sporulating forms are not destroyed here. They are killed here, or their number is reduced so that the load becomes less. The number of microorganisms is reduced here, so that they are not causing any harm, they are not causing any danger. Number is very less there. Generally, these disinfectants are chemicals. Okay, and these disinfectants basically they are used on inanimate objects, not on living tissues, and these basically are used as surface disinfectants like your Lysol, etc. They are not used to kill resistant spores. Moving on to the next term, that is a germicide. Germicide is a term which we can use which will kill the vegetative cells, but definitely not spores. Okay, and germicide can act as a disinfectant and also can act as an antiseptic. These germicides basically are used to destroy or get rid of the pathogens. Germicides are narrow spectrum, wherein it could be either bactericide, wherein it destroys the bacteria, pathogens, which is bacteria, or pathogenic fungi, etc. So, they are narrow spectrum. But when we talk about disinfectant, they are getting rid of microbes. Load is becoming less there on inanimate objects. Okay, germicide, it could be on animate objects if it is an antiseptic, and it can act as a disinfectant also. But basically pathogens are destroyed here. Now here, how are they? They cause severe injury to the microbes, okay, and they will not be allowed to multiply further. Moving on to the other terms, what we have to study is microbiostatic, microbiocidal and microbiolytic. What do you mean by microbiostatic? Is I am stagnating their growth. I am not killing them, nor am I allowing them to multiply. Whichever phase they are in, the growth just becomes static. And also here, once I remove the chemical from that particular, or I remove that agent from that particular surface, from that particular part, the organisms which have become static, which have become stagnated, the growth revives. They can get revived here. Microbiocidal is, I am killing the organisms probably by physical methods, or I use certain chemicals wherein I am killing the microorganisms. It can be bactericidal, fungicidal, etc. Microbiolytic. Now here also I am killing the microorganisms, but I am killing by lysing the cell. The cell gets lysed here. Basically it is used for bacteria. These chemicals are used, or these agents are used to kill bacteria by lysing. Okay, here basically the cell wall or the plasma membrane gets lysed and definitely the cell gets destroyed here. So that is lytic. Cidal is, microorganisms are killed by any methods, but here they are killed by lysing the cell. So these are the different terms which we had to study before studying sterilization. So sterilization, wherein it is zero organisms. Sterilization, I can heat the food so that the number becomes less, but I am not sterilizing the food. So that is a difference that you have to remember there. Okay. So thank you.